The history of the fluid mosaic model. The plasma membrane structure was explained through the discovery of the fluid mosaic model in 1972. This was discovered by S.J. Singer and Garth L. Nicholson. The model sticks to the basic principle of how the plasma membrane structure and its functions are. No matter how the fluid mosaic model evolves from time to time, it still explains the basic same general idea of how the fluid mosaic model is arranged. The fluid mosaic model explains how the fluid characteristic of the plasma membrane was gathered through the mosaic arrangements of the plasma membrane's component that includes phospholipids, cholesterol, protein, and carbohydrates. The model is supported through the evidence from different areas. One of those evidence is the prediction on the enzyme-like transmembrane protein to the studies of permeability and transport. Another evidence that suggested the fluid mosaic model is the participation of proteins in the membrane plane that was noted due to the apparent lack of lipids and to make up complete bilayers. Pictures of electron microscopy suggested the presence of proteins within the membranes. These are just some of the scientific evidence that supported the structure of the fluid mosaic model.
Plasma membrane components are lipids, which include phospholipid and cholesterol, proteins and classes of carbohydrates that attach to some lipids and proteins. Okay, first of all, the first main component is phospholipid, where it is arranged in bilayer and it's the essential structure of plasma membrane. It is also amphithetic, meaning that it has hydrophobic and hydrophilic parts, and which hydrophobic means that it's a water-loving part, which includes of negatively charged phosphate group and also a small group that has a charge. Hydrophobic part is water repelling state, which is made up of non-polar fatty acid and also interacts well with non-polar molecules and doesn't react well with water. So second main component of plasma membrane is protein. It has two categories, which are integral membrane protein and peripheral membrane protein, where integral membrane proteins are incorporated with plasma membrane itself, and it has at least one hydrophobic region which anchors into phospholipid bilayer. Proteins which extend throughout the membranes are called transmembrane protein. Some of integral protein membrane forms a channel to pass the small ion toward the plasma membrane. And the second is the peripheral peripheral protein found outside and inside of the membrane that are attached to the integral protein or phospholipid. They always appear to attach loosely. The last part of the main component of plasma membrane is carbohydrate, which found on the outer surface of cells and bounded to proteins, which are called glycoproteins, and also lipids, where it is called glycolipids. It forms a distinctive cell markers which allow to recognize one another, where the markers are important in the immune system.
There are a few reasons that leads to membrane fluidity. First is the mosaic characteristic. The integral proteins and lipids exist as separate but loosely attached molecules. Second is the phospholipid molecules. In their saturated forms, there are no double bonds between the adjacent carbon atoms and therefore result of a straight tails, opposite of the unsaturated fatty acid, which have a double bonds that result of the bend of approximately 30 degrees in the string of carbons. This is important in the environment as it tends to compress the membrane, as it is composed of mostly unsaturated fatty acid, thus making them less fluid and susceptible to rupture. On the other hand, if the unsaturated fatty acid are compressed, the kin in their tail elbow adjacent phospholipid molecules away, maintaining some space between the phospholipid molecules. These elbow rooms will help in maintaining fluidity in the membrane at temperature at which membrane with saturated fatty acid tails would freeze or solidify. For example, fish are capable of adapting to the cold surrounding by changing their proportion of unsaturated fatty acid in the membrane in response to the low temperature, alongside the phospholipid in the membrane, and tends to dampen the effect of temperature. It prevents the low temperature from stopping the fluidity of the membranes and prevents the high temperature from increasing the fluidity of the membrane too much.
For my part, let me explain about the diffusion through lipid bilayer. Each and every living cells must be able to exchange materials, either end products or nutrients, with their external environment so that they can stay alive. It is crucial for us to understand how different types of molecules can pass through the lipid bilayer, since the phospholipid bilayer is responsible for forming membranes and compartments. Biological membranes are semi-permeable lipid bilayers. It means that it only allows certain types of substances to pass through the membrane. Let me explain about permeability. Permeability means the tendency of each molecule in passing through the biological membrane. Lipid-soluble molecules and certain small molecules are able to freely pass through the lipid bilayer. It is due to the chemical and structural nature of the phospholipid bilayer. In addition, to determine whether a molecule can diffuse through the membrane, it relies on the size of the molecule and its electrical nature. The membrane is highly permeable to non-polar, which are fat-soluble molecules. The permeability of the membrane to polar molecules is extremely low, whereas large polar molecules, they cannot diffuse through the membrane. The permeability to ions, which are charged molecule species, is particularly extremely low as well. Hence, the molecules and ion pathway is facilitated by the existence of specific membrane transport proteins, which are carrier proteins and channel proteins.
Ions such as sodium, potassium, calcium and chloride are important for many cell functions. Because they are charged polar, these ions do not diffuse through the membrane. Instead, they move through the ion channel proteins. Ion channel allows the formation of concentration gradient between the extracellular fluid and cytosol. Ion channels are very specific as they allow only certain ions through the cell membrane. Some ion channels are always open, others are gated. Gated ion channels can open or close in response to different types of stimuli such as electrical or chemical signals. They are said to be gated. Some types of gated ions are ligated gated, mechanically gated, voltage gated and light gated. Ligand gated ion channels are open or closed in response to binding a small signaling molecule or ligand. Some ion channels are gated by extracellular ligands, some by intracellular ligands. And then the external ligands. External ligands bind to a site on the extracellular side of the channel during the diffusion of the ions. Following it, the internal ligands. Internal ligands bind to the site of the channel protein exposed to the cytosol. Finally, the patch clamp technique. The properties of the ion channels can be studied by the means of patch clamp technique. A very fine pipette with an opening of about 0.5 micrometer is pressed against the plasma membrane of either an intact cell or the plasma membrane can be pulled away from the cell and preparation placed in the test tube solution of the desired composition. Current flow through a single ion channel can be then measured.
Facilitated diffusion is another type of passive transport across the cell membrane. As per the name, it relies on the concentration gradient of the substances, will flow from a high concentration to a lower concentration. It occurs spontaneously, involving substances that typically cannot pass through the phospholipid bilayer through normal diffusion, such as large polar ions. Similar to the ion channels, facilitated diffusion also involves a protein to add the substances to go through the membrane. This is because of the hydrophobic nature of the bilayer that does not allow larger polar substances to pass. In facilitated diffusion, the particular integral transmembrane protein involved is the carrier protein. Unlike channel proteins, which simply provides a pathway for the ions to move through, carrier proteins change shape when the substrate binds to it in order to transfer it to the other side of the membrane. Carrier proteins may also be involved in active transport, but there are specific carrier proteins solely for passive transport. This means that they do not require the chemical energy from ATP hydrolysis for it to occur, as long as the concentration gradient is present. There are three main types of carrier protein: uniport, symport, and antiport. A uniport only allows one specific type of substances to pass through the membrane, while a symport allows two types of substances at a time at the same direction. In contrast, an antiport transports two types of substances as well. However, those substances move in opposite directions and occur simultaneously. They are also known as co-transport proteins. Where proteins are only open at one side of the membrane at a time.
Active transport is the process of moving materials across the cell membrane against its gradients, and it requires the use of energy. Ions move from low concentration to high concentration. Active transport mechanism expending energy usually in ATP form to maintain the concentration of ions in living cells. Transport mechanism can be divided into several types. First is primary active transport. Sodium-potassium pump is the example of primary active transport because it uses ATP as its energy source to move the molecules across the cell membrane against the gradient. It transports sodium ion out and potassium ion in to the cell in a repeating cycle. For each cycle, it pumps 3 sodium out and it will pump 2 ions enter the cell, and the ATP will allow the shape of the pump, which is carrier protein, to change. Next is endocytosis, another type of active transport. In the previous example, which is sodium-potassium pump, the molecule used is small, but in endocytosis, energy is used to take in biological molecules and large particles by the formation of new vesicles. There are multiple types of endocytosis, with major categories being phagocytosis, phagocytosis, and receptor-mediated endocytosis. Next is exocytosis. Exocytosis is opposite of endocytosis. In exocytosis, the vesicle fuses with the plasma membrane, thereby releasing all the content and waste out of the cell. This type of active transport is mainly used by secretory cells where they secrete insulin or neuron.